I'd like to invite Christina Giannakou. Christina was born in Greece, but she's currently working on a PhD project in the Netherlands. It's a joint project from the University of Maastricht and the RIVM, the National Institute for Public Health and Environment in Bilthoven. And she's going to present actually the most important outcomes of her thesis. Christina.
Thank you, Robert. Good afternoon from me too. I'm going to talk about, part of my PhD project and present an integrated testing strategy for immunotoxicity testing of nanomedicinal products. Really quickly, a background how on the immunotoxicity testing. So nanomaterials, but also nanomedicinal products, are reported to have a different toxicokinetic profile compared to conventional drugs. So they tend to accumulate in the organs of the immune system, where they are able to interact with it. And these interactions result in potential side effects with different intensity and extent. So the immunotoxicity profile, so the safety evaluation of the immune system for the nanomedicinal products is of high importance. Taking this as a beginning step, the aim of my project was formed, and in order to approach it, 3 objectives were set. The first one was to compare the present guidelines, with the increasing knowledge on the potential immunotoxicity assessment of nanomedicinal products in order to identify gaps in the safety evaluation process. The second one, after identifying these potential gaps, investigate if in vitro assays currently available are can be effectively used to assess potential immunotoxicity of these products. And finally, based on the information gathered in the 2 objectives before, create an innovative testing strategy for immunotoxicity testing. I will focus mainly on this objective and the outcomes of this project, and start with a small background on the regulatory framework. So we heard several information for safety evaluation of nanomedicinal products before. I'm going to talk on the immunotoxicity testing, and the regulatory documents. The guidelines that are currently used are guidelines designed for conventional medicines. The ICH-8 is the guideline used currently for medicinal products. There are 4 reflection papers developed from the European Medicines Agency addressing and actually specifically in one of them, immunotoxicity is being addressed in a bit more detail apart from what is being incorporated in the ICH-8, or maybe some guidance can be shared in ISO documents, for example, this one that is for medical devices. However, there are no regulatory guidelines that are specifically dedicated on immunotoxicity of nanomedicinal products. We screened, so taking the ICH-8 mainly and the testing assays that are being proposed within this guideline and the testing techniques that are being proposed in literature, we tried to compare, we tried to see if the endpoints, if the adverse effects found in the immunomodulatory effects, detected in the literature after administration of nanomedicinal products or nanomaterials, for example, immunosuppressive effects, immunostimulative or hypersensitivity effects, either in vitro or in vivo, are able to be readily detected by the assays found in these 2 categories. So what we saw is that most of the testing assays proposed by the guidelines are not able to readily detect adverse effects found either in clinical studies or in vitro tests by the non-clinical tests proposed by the ICH-8. Next one. Yeah. So. Establishing the gaps found after this comparison, we formed an integrated testing strategy, which is summarized in 5 parts in this slide, starting with factors that we can identify either for either factors that should be taken into account that they trigger adverse immunotoxic effects, or factors that can be used to to design the further non-clin in vivo studies. We continued with a part on the in vitro assay cascade. The information of these two parts will be taken into account, and any weight of evidence review will be composed. Based on expert judgment, further in vivo tests will be decided, and the information of all the four parts will contribute to the conclusions, which will be taken along in the risk assessment or give input in the clinical studies that follow. In a bit more detail, the first part, so the factors to consider, we split them in two different parts. First of all, factors that trigger immunotoxicity testing, or they can be taken into account the study design. And there are factors, the ones in blue, that are already present in the guidelines that we used as a basis for our strategy, the Assay, or factors that they are displayed in green, that they are factors we recommend that should be included for immunotoxicity testing specific. One of the factors we use, the pharmacological properties, is a modified factor of a factor that is present in the ICH Assay, which gives in detail that pharmacological properties should be included for the whole product, but we propose that of each part of the nanomedicinal product should also be information for their pharmacological properties. As well as physicochemical characteristics should be included, and information on the endotoxin presence. One of the factors that is being proposed is the route of administration. And based on the information and based on the route that is being used, the further assessment of the material will be decided. So, in case there is dermal administration, there can be either a systemic exposure or not. So, in case systemic exposure can be established, further testing, like materials administered from any other route, can be used. But if there's not a systemic exposure, then further immunotoxicity testing should be considered for sensitization. The second part is comprised from the in vitro testing cascade with the following 6 parameters: the complement activation, macrophage function, myelosuppression, lymphocyte function, inflammasome activation, and DC antigen presentation. And for these parameters, the conditions that should be taken into account while testing is that the assays, so the readout system chosen to address these effects, these parameters, the predictability of these assays should be taken into account, and pitfalls, including interference of the product tested, is of importance and should be readily tested for with appropriate controls. So the third part is taking information from the first two parts, and based on the expert judgment, the type and the design of the in vivo studies will be decided. And after conducting these in vivo studies, the conclusions will be drawn to go further for the design of the first clinical studies and the risk assessment of the material. So this brings me to the conclusions of these parts. It is clear, you saw that it's clear that an NMP-specific testing strategy for the safety of the immune system is needed for nanomedicinal products, and here we suggest an integrated testing strategy where nanospecific factors, but also in vitro tests are part of it. And I will close with some recommendations. The strategy is formed as an integrated testing strategy, which can be part of an IATA, so an integrated approach, where information from which are being used for chemicals. They start being used for chemicals, where they use also AOPs or modeling to help in the risk assessment. Something like that is still in the infant stage for nanomedicinal products, but they can be taken into account when they are more developed, and they can give input in the assessment for immunotoxicity of nanomedicines. The integrated testing strategy and the information yielded from it can give input in the safer by design concept, which we heard Robert some further information before, or they can be used in the read-across concept to help in the assessment of newly developed drugs. And finally, maybe grouping maybe can contribute in further grouping of nanomedicinal products. And with this, my time is ended, and I would like to thank everybody that contributed in this project, and leave you with a take-home message that this immunotoxicity is crucial, and integrated testing strategy is needed for these products. Thank you very much for your attention.
Thank you very much, Christina. We have room for a few questions. Susanne?
With regard to your strategy, when you come to an in vivo test, we know that the immune system is highly interspecies variating. How do you deal with that, and how do you address that?
Well, for the in vivo tests, there is quite an extensive. There are quite extensive parameters proposed already in the ICH-8, mostly rodent models. But there is already knowledge that for some endpoints, for example, CARPA, a pig study would be more appropriate. So the correct model should be chosen to be able to test for the endpoints for the parameters needed. So based on the accumulating knowledge, it's not like you said, it's not a universal. You can't use one specific animal model or one specific species. So depending what you're testing, you choose the corresponding one.
Could you speculate on your final slide on the moving forward on the aspect of the XR? Because XR are very structural activity related to manufacturing material, and the read-across aspect as well, in a context of nanomaterial using medical devices.
Medical devices? Yeah. I'm not really, I don't have a lot of extensive knowledge in medical devices. I can say some things about nanomedicinal products, and maybe this can help. So QSAR models, they can help give you some information how your particle can react, or they can predict how it can behave in different media. So they can maybe help you choose in vitro models, and during the screening or the in vitro assay cascade, help choosing the best readout to address the specific parameter that needs to be addressed. This I guess can also be used as a parameter for medical devices, or I don't understand your question correctly.
Just to elaborate, where in nanomedicine you have a dispersion, so you can actually look at the structural activity in a media. Yeah. In a material which is encapsulated as a trap, the structural activity is functional of the entire matrix. That was just divided into two aspects, where in one case you need to decouple what is the material nano versus the medical device and so on. What about the read-across? Because also read-across has been developed in a different context, if I may ask the question. But then,
yeah, there are some first attempts in read-across concepts for nanomaterials in general, and hopefully the information yielded can help in the assessment for the nanomedicinal products. But they can maybe help in the grouping, so more also in the to predict the behavior in the body, to predict also the adverse effects that may have, because maybe not all of the materials have all the adverse effects that we were able to identify.
Okay, thank you very much.
Thank you.
One brief remark at the end. Unfortunately, in view of the time, there was no occasion for Christina to talk about the large experimental work that she's been doing. But part of the tests that Christina has been working on will be also worked on in a European project. We have several partners and even a coordinator in the room. The project is called Refine. So you may hear more about her, at least the next steps in the experimental work that she has been doing in the first already.